of our team transporting the pig kidneys. You can see Dr. Arandi is holding a styrofoam cooler. That is where the pig kidneys were contained. This is similar to the practice we have in human-to-human allotransplantation. In this picture, you can see the pig kidney, and this is prior to restoring blood flow. You can see it's pale. We then transplanted and completed our vascular connections and restored blood flow to the kidney. You can see in the next picture that it's beautiful in pink. That's a Doppler probe listening to Doppler signals throughout the kidney. And within 23 minutes of restoring the recipient's blood flow to the pig kidney, it began to make urine. This is a remarkable accomplishment. It tested from start to finish how we might recapitulate this in a living person, testing both key safety and feasibility measures. One of the key things that needed to be done was the development of a flow crossmatch that was specific for pig to human transplant. Flow crossmatches are used in human-to-human transplantation to ensure that there is tissue compatibility, meaning that when we transplant that kidney, that it will not immediately hyperacutely reject, meaning that it would reject right before our eyes and have to be explanted. In fact, this is considered such a key element of transplantation that federal regulators require crossmatch testing to be completed prior to transplantation in the context of human-to-human transplant. Prior to our study, no such test existed, and I want to give tremendous credit to Dr. Hopfeld and her team in our histocompatibility laboratory who developed this novel flow crossmatch that was specific for pig to human transplant. They were able to predict a priori, meaning prior to transplant, that there would be a negative crossmatch, meaning there would not be hyperacute rejection. The only way to test that was to do the transplant and show that after reperfusion, we had a beautiful pink kidney, not one that turned black from hyperacute rejection. This crossmatch only exists here at UAB, and it will need to be leveraged for the widespread adoption and use in living people for the purposes of pig-to-human transplantation. As I mentioned, we also test the process from start to finish. We also were able to assess the recipient's blood for the presence of pig viruses, and we were able to ensure that we did not transmit any porcine endogenous retroviruses, again confirming our ability to maintain a pathogen-free facility and to have the safe procurement and transplantation of pig kidneys into humans. We also were able to demonstrate that we did not observe any pig cells circulating in the recipient's blood. Another important milestone, we could be reassured that we were not going to see graft versus host disease, which can in fact happen after transplantation, even in human-to-human transplant. And I think importantly, beyond xenotransplantation, this study really established brain death as a preclinical human model to study the human condition. And I just cannot thank the Parsons family enough for honoring Mr. Parsons' decision to be an organ donor upon his death. And he was agreed to be an organ donor both for the purposes of transplantation as well as research. It was important to him and to his family to honor his legacy by being able to give back to the community around him and to help people he has never met. What Mr. Parsons did and the decision that his family honored has not only helped patients who have kidney failure one day be able to achieve a transplant where we eliminate the organ shortage crisis. There's no longer a wait. When you need a kidney, we have one. But beyond that, this model, I believe, will be able to be leveraged to study all sorts of human conditions in which animal models are not appropriate. And the Parsons family and Mr. Parsons deserve all of the credit. And we propose heretofore that this model be known as the Parsons model. And with that, I do want to thank the team both at UAB Medicine, the Legacy of Hope, as well as the UAB Heersink School of Medicine for all the hard work. This picture represents the individuals that were involved in the operating room, but it doesn't do justice to all the individuals that were involved in this process, many of whom have joined us today and are in this room with me as a part of this press conference. To each of you, there are too many to name, but we say thank you. And on behalf of our patients, we say thank you. Today, we just gave hope to the hundreds of thousands of Americans who suffer from kidney failure, who don't have access to a life-saving kidney transplant. To the Parsons family, thank you. We wouldn't be here without you. And for all of our patients, and on behalf of all of them, we say thank you. You have saved countless lives moving forward, and I hope that gives you solace in your grief very much. And we will be answering questions at the end. At this time, I would like to introduce the Chief Scientific Officer for Revivicor, Dr. David Ayers.
Dr. Ayers, we cannot hear you because you're on mute, so we're going to ask you to unmute now and start over, please.
Thanks, Dr. Locke. This is indeed an exciting day. All of us at Revivicor, along with our parent company, United Therapeutics, are in awe of the historic achievements at the University of Alabama, Birmingham, with our investigational 10-gene U-kidney. The U-kidney may turn out to be a life-saving solution for thousands of people on dialysis, subject to successful completion of future clinical trials and achievement of full FDA approval. This preclinical study and resulting peer-reviewed publication will assist in the further development of xenotransplantation. Revivicor and United Therapeutics are committed to continuing our development efforts with the goal of FDA approval of our xeno organs like U-kidney to address the critical unmet need for transplantable organs in the United States. We carefully selected and tested 10 gene edits reflected in the U-kidney to facilitate compatibility with the human immune system. Building on the GalSafe gene modification, this is the first FDA-approved genetic modification in a mammal. The donor pig had 10 genetic modifications, including the knockout of 4 pig genes and the addition of 6 human genes. These modifications may make the pig organs more tolerable for human transplant recipients. Additionally, thanks. We stress that this historic achievement is an experimental step, part of the process that we hope will lead to securing approval for use in patients. The U-kidney is not approved by the FDA, and we, along with our academic collaborators, will continue to pursue our development and regulatory path towards obtaining FDA approval. The recent procedures by UAB and those reported by Revivicor and other United Therapeutic collaborators over the past few months represent the culmination of 2 decades of work by my team, starting with the first cloning of pigs to optimizing various gene editing constructs and building state-of-the-art farm facilities to house these incredible animals. We look forward to continued engagement with FDA in the coming years to bring engineered organs like U-kidney to patients in need. So turn it back to Jamie. Thank you.
Great, thank you, Dr. Ayers. It's now my distinct pleasure to be able to introduce Mrs. Ohara. Mrs. Ohara is...
Dr. Locke, we're having a few technical difficulties. If you could hold for just 1 second, please. This microphone is muted. If the production team can unmute. Okay, Dr. Locke.
Partner in rearing their children. And was
Dr. Locke, we're going to have you hold for 1 more moment. I apologize again. We've got to figure out what's going on with our sound quickly. Just can you hold for just 1 second, please?
Sure. Good morning. Can you hear me? Okay. Again, I just want to say it is my distinct privilege and honor this morning to introduce Miss Ohara, who's the ex-wife of Mr. Parsons, but importantly, his longtime partner, as they describe it, rearing their children, and importantly, being a critical component of making this decision and helping to honor Mr. Parsons' legacy. So, Ms. Ohara, thank you for being here, and I'll turn the podium over to you.
Good morning. Thank you for being here today to learn about what we believe is a remarkable scientific achievement and to learn about and celebrate the life of a father, a son, a friend, and a servant to others, Jim Parsons, a man who truly lived to help others in both life and death, as evidence of why we are all here today. I'd like to share a bit about Jim so people will know what type of man he was. Jim was fun. He lived life to the fullest, ready for the next great adventure. He lived for his family, namely his children. He lived for curvy roads on two wheels and tight woods with muddy trails. By day, he was a carpenter. He could transform your kitchen or give you the closet of your dreams. But at the very core of Jim Parsons was his love of riding motorcycles. He started at the age of 14 and developed into a great dirt bike rider, winning countless first-place trophies. This is ultimately what took his life. But before he died, just 2 weeks before, he took his sons and my brother, his best friend Ryan, on a trip of a lifetime to ride dirt bikes in the mountains of Colorado. He took them to all of the places he'd always dreamed of showing them. And it was when they got to the top of American Flag Mountain that he confided to the boys that this was where he wanted his ashes spread. Just a couple of weeks later, a few days after his 57th birthday, Jim was racing with our son David and my brother Ryan like he had done hundreds of times before. He knew the potential consequences of racing. Every racer does. But that day, he hit a tree the wrong way and succumbed to those consequences. He was resuscitated and life-flighted to a hospital in Montgomery, but we were quickly informed that he was brain dead. We had time as a family to grieve and to say our goodbyes, but we also knew that Jim had more to give. He and I had often talked about being organ donors. His parents were leaving their bodies to science, and we agreed that organ donation was what we wanted to do upon our death. So when we were approached by Legacy of Hope and Dr. Locke about this study and what she hoped it would ultimately accomplish, we had no doubt as a family that this is what Jim would have wanted, to potentially help hundreds of thousands of others. If he knew that participating in this study could potentially give the nearly 100,000 people in the United States waiting for a kidney transplant the chance at living a healthy life, he wouldn't have had a moment's hesitation. He would be all in. That was who Jim was. Our greatest hope is that this study, which is now called the Parsons Model, will ultimately bring new life to hundreds of thousands of people in the United States and around the world in need of a kidney transplant. We also want to bring awareness to the need for organ donation and share light on how big of a crisis organ shortage is. Our daughter, Allie, said recently that losing her dad is the hardest thing that she's ever been through. It still is, and we are reminded of that every day. But we know that Jim is so proud that we are gathered here today and that we have had the opportunity to share his story and his legacy. More importantly, we hope our story inspires other families to consider organ donation and sharing the gift of life forward as Jim did. On behalf of Allie, David, Cole, and our entire family, I want to thank Legacy of Hope and UAB, especially Alan Spriggs and Dr. Jamie Locke. Throughout this journey, they have been Jim's advocate. And we thank you all from the bottom of our hearts. And all of you out here, it's just overwhelming to see the number of people that it took to put all this together. We all look forward to the day when the first pig to human kidney transplant takes place in a patient that can go on to live a long and healthy life. We're forever honored that Jim's gift has an opportunity to give those in need a chance at life. Thank you.
Thank you, Julie. We're going to ask Dr. Lott to join and Dr. Vickers to join Julie up here for questions. Media, if you have questions, please, Savannah Copeland from our office will be calling on you. Just raise your hand in the Zoom function or drop a question in the chat and we'll get started.
Thank you all for joining us. As Tyler mentioned, if you have a question, please use the raise hand function or drop your question in the chat and we will ask on your behalf. Please state your name and your outlet, and if you have a question directed for a specific speaker. In an effort to not cause any further issues with audio, any questions directed for David with Revivicor will be arranged by phone after the press conference. So with that, we will open. The first question is from Francis Sellers with the Washington Post. I think, Dr. Locke, this will be for you. Can you comment more about how this is a first? Is it because the research was peer-reviewed and published? What specifically sets this research apart from others in the news recently?
Thank you for that question. I think there are several notable firsts that make this the first. The first thing is that this was a clinical grade 10 gene edited pig that was transplanted inside a human recipient in the same fashion that we perform human to human allotransplantation. When I say clinical grade, it means that the pig kidney came from a pathogen-free facility, and so that makes it a first as well. In addition to that, it's the first time that a pig-to-human specific flow crossmatch has been developed and then actually validated. So we predicted with a negative crossmatch that we would not see hyperacute rejection, but the only way to validate the crossmatch was to actually perform the transplant. And as you could see from the pictures, the kidney was beautiful and pink and did not hyperacutely reject. We also were able, for the first, to demonstrate that there was no transmission of porcine endogenous retroviruses in the human recipient. There was also no evidence of chimerism, meaning we did not detect pig cells circulating in the human recipient's blood. And yes, in fact, this is peer-reviewed, which also makes it a first, as all of these data have been critically reviewed by other scientists and experts in the field, confirming our findings and now available online via the American Journal of Transplantation. The other thing that this did is really test operationally our ability to do this from beginning to end. So you have to visualize the concept of having a pathogen-free facility with an operating room that meets standards that you would see in a human hospital, JACO-type requirements, where these pig organs could be harvested in a sterile fashion, packaged, transported to a hospital, and successfully transplanted into a human brain-dead recipient. That really tested key operational points that are going to be necessary if we want to extend this beyond an individual patient and be able to offer this to the masses. And I think each of these firsts that I described are going to be key data elements that federal regulators are going to want to see and understand that could not be answered in the context of a non-human primate model. So we're very excited about this because we genuinely believe this is going to be what it's going to take to help us advance and move the field forward. Thank you.
Thank you. Ilya Kolmanovsky, you can unmute and ask your question. Please state your affiliation.
Hello, this is a Naked Mole Right podcast, and this one is probably for Dr. Vickers. Could you please give us some timeline of your collaboration with Revivicor? Who initiated it? Who reached out to whom? How did it start and when? And did that FDA approval of alpha-gal safe pig in December last year play any role in that timeline?
So as Dr. Ayers mentioned, we've had a long-standing relationship with United Therapeutics. Their CEO, Martine Rothblatt, has been a tremendous collaborator for many institutions in this field, but particularly with UAB over the last really 3 to 4 years. That partnership really has driven the multiple both studies, both in research and clinical, for us to move to this level. So I think, as you heard initially, really our 4-year culmination of work with us as one of the main institutions. The concept of this has been around some time, but I have to say that Dr. Locke was one of the first to propose that this could really give us information that could lead the way to a clinical trial for transplantation. And the evolution of the editing of the pigs and the genes and CRISPR-Cas obviously had a huge impact on creating the opportunity where these organs would not be rejected. I didn't quite understand your question around December. You were asking about...
There was this milestone. Yeah, there was this milestone December last year when FDA... By the host now, sorry. Let me repeat it. December last year, FDA has approved gall-safe pigs by Revivicor. I guess to be eaten, but it was in small print for therapeutics as well. I was wondering if this was a milestone for moving forward with xenotransplant. But
Yeah, thank you for that question.
was this a milestone for you?
I think certainly the approval of the gall-safe pig, which again was done for the purposes of food consumption, is certainly a milestone, certainly for Revivicor, but also for the broader field of xenotransplantation, because anytime we have the opportunity to interact with the FDA, be able to demonstrate data, and be able to gain approval, I think that does move the field forward. We're going to need the FDA's approval of these genetically edited pig kidneys in order to move this into living persons. So I think that certainly was a nice step along a very long process that's going to require further interactions, but certainly I would consider that a milestone, yes.
Thank you.
Okay. And for the future, sorry.
Sorry, we've got another question.
I'm wondering what...
Sorry, feel free to keep going.
Oh, thanks. I was wondering what's the roadmap for the future? How long until the actual xenotransplant in human, and what are the main milestones to be achieved?
Thank you for that question. You know, our hope is to continue to move this process along. I think the data that has been collected from this particular study is going to be key as we begin to have discussions with the FDA about obtaining an IND, which is something that we would like to do. We also have been working with our internal review board about what a phase 1 clinical trial would look like. And certainly having IND approval from the FDA will be a necessary component to receive approval from our IRB to move forward with a phase 1 clinical trial. We are very hopeful that we are able to get an investigator-initiated IND at some point this year, and we are very hopeful that we would be able to implement a phase 1 clinical trial later this year. We do have a herd of pigs that will be ready in the coming months in terms of appropriate size that are 10 gene edited for us to begin the phase 1 clinical trial. So my hope is that sometime later this calendar year.
Thank you. Julie, the next question is going to be for you. A few people are asking just a little bit more about what that process of making this decision was like for your family. And through this, how do you and your family want to memorialize Jim?
So, as I said, we were going to donate Jim's organs. And about an hour and a half before his surgery to harvest all of his organs, I got a call from Legacy of Hope. And Dr. Locke got on the phone, and my daughter and I were in the car, and she put the whole thing out there and what was going to happen if we agreed. And my daughter and I looked at each other, and we were just, without a doubt, we knew that that's the way we wanted to go with Jim. So we halted his surgery, and UAB arranged to have him transported up here from Montgomery, which was where he had been life flighted to. And then the second part of your question was?
How do you hope that this process memorializes him moving forward in his memory?
We are just thrilled that they've decided to call this the Parsons model. And I think he would be very proud also. He would probably be saying, stop talking about me so much right now. Let's get the focus on all of you people in the room here and off of him. But we're very proud to memorialize him this way.
Thank you. Dr. Locke, the next question will be for you. You touched on this in your presentation, but a few people are just curious. Can you talk a little bit more about the crisis of kidneys being in need, and what you hope this means for the future of those patients who are on a list?
Absolutely. So the thing about kidney failure is I think many people sort of say, well, we've got dialysis, we're good. But dialysis is awful. It comes with a significant mortality. As I mentioned, 40% of the patients on the waiting list, if they don't get an offer and actually get transplanted, will die in 5 years. And so dialysis is not the answer. Since Joseph Murray performed the first successful kidney transplant, I believe in 1954, for which he won the Nobel Prize, we have known that kidney transplantation, in fact, is the gold standard, and it is, in fact, the cure for end-stage kidney disease and kidney failure. And so imagine any other disease condition in which I, as a physician, along with many of my colleagues in this room, I've got a cure. I know exactly what the cure is, and I don't actually have enough to go around. And now imagine every week, and we have evaluation clinic at UAB 5 days a week, and we see patients every single day, Monday through Friday, and we evaluate them for waitlisting. And every day, myself or one of my colleagues has to tell someone that either they're not a candidate, or if they are a candidate, they're going to have a really long wait. And depending on their blood group, the wait could be upwards of 10 years. And many of these individuals are going to die before we ever get them a kidney. We have tried so many things in this country to increase organs, the availability of organs, and a huge shout out to our organ procurement organization, who's one of the best in the country at doing it. Their conversion rate is fantastic, but yet the need still far exceeds our supply. So if you look right now in the U.S., there are a little under 100,000 individuals waiting for a kidney transplant, and this year we'll perform fewer than 25,000. And if you back it up further, there are another 600,000 Americans that actually have end-stage kidney disease that have yet to make it to a waiting list. And oh, by the way, there are 37 million Americans that have chronic kidney disease. Many of them don't even realize they have it, but will go on to progress and develop kidney failure and ultimately need a transplant. Again, they have a disease that we have a cure for, but we don't have enough. And we need a radical solution. We have to find an alternative organ source, and xenotransplantation, I believe, is the answer. And it is my great hope that certainly in the next 5 to 10 years, and if not by then, during my lifetime, that xenotransplantation becomes a reality, so that every patient that walks into our clinic that has this need, that my colleagues and I can give them their solution. And I think what we have done here today, I hope, if nothing else, in the immediate, provides hope to all of those individuals who are waiting, who are looking death in the eye and are hoping beyond all hope that they get that call for a transplant. I hope this gives them hope for a future where there will no longer be a wait.
Thank you. Dr. Vickers, the next question's for you. When this procedure becomes possible, will UAB have to grow its infrastructure to make this work as well as it could?
We would love to have that problem. And as you've heard from Dr. Locke, the burden of disease for chronic renal disease is huge across our country. And as you heard me mention earlier, we sit in the epicenter of that, where the greatest burden exists. And we've been committed to serving the populations of this region throughout our entire history with what the latest science had to offer, as well as helping define that science. And so, yes, you can imagine that we would need a specific facility. We would need newer, in part, more individuals to help drive it. But it's a graduated process. The phase 1 clinical trial has to be done first. We have to be clear about the right model for the pig to make this happen. And then we need to make sure we have the recipients who are the right ones to get the organs. And if we were to progress through that process, then we would love to be able to upscale and grow. And we have our leaders of our health system and our hospital here as well, who would love to take on the task of making that a reality for our surgeons, but our patients and our citizens.
Dr. Vickers, actually just another thing to ask you. Can you just speak a little bit more about what does this mean for UAB to be at the epicenter of this research?
You know, I go back to Dr. Watt's leadership, who's pushed us to really, at every level of our institution, particularly on our academic medical side, to really be a leader in the country and the world for the areas that we know that we can do that in. Transplantation is clearly one of them. You heard me list that unique history of beginning a transplant program, one of the earliest in the world, and albeit in Alabama, we did it and have grown it to be one of the biggest in the world. So it's just a reflection, as you've heard, that this did not occur overnight. This has been a long journey and commitment of our institution to actually be an outstanding academic medical center that any place in the country could be proud of, and provide the latest opportunity, whether it's for cancer, cardiac, or transplantation and medical care, that would not only benefit citizens here, but would benefit people for all over the world. So it's a reflection of who we are and what we've wanted to actually demonstrate throughout our history.
Thank you. Dr. Locke, can you comment a little bit more? Are these bridge to transplant kidneys, or will these be usable long term?
That's a great question, and I think one of the things and reasons why we need to be able to progress and move this into living humans. Our hope is that this will be destination therapy, meaning the patient receives the transplant and it survives the duration of that recipient's life. But we need to do the research to determine that, and that's why we need to be able to progress to a clinical trial in living persons. I do think it's important to note that the lifespan of a pig is about 30 years, so it is our great hope that we will be able to make use of that full lifespan such that a pig kidney could sustain a living person dialysis-free for 30 years.
Thank you. Tyler, I think that's all for our questions.
All right, thank you everybody for being with us today, and again to members of the media on the call. As a reminder, our institutions, University of Alabama at Birmingham on first reference and UAB on second, and you can also get the media kit at go.uab.edu/xenotransplantation. Thank you very much. We appreciate you being here.